Cancer. It's a word that strikes fear into the hearts of many. But what exactly is it? At its core, cancer is a genetic disease. It's a disease that arises from changes, often subtle and unseen, within the very blueprint of our cells, our DNA. These changes, known as mutations, disrupt the carefully orchestrated process of cell growth and division, leading to uncontrolled proliferation and the formation of tumors. Imagine our DNA as a vast library filled with volumes of instructions. Each gene, like a single volume, contains the code for building and maintaining our bodies. When these genes are altered or damaged, the instructions become garbled, leading to errors in cell behavior. This is where the story of cancer begins. These genetic changes can occur throughout our lifetime, often as a result of mistakes during cell division or exposure to harmful agents in the environment. While our bodies have remarkable repair mechanisms to correct these errors, sometimes these safeguards fail, and the altered genes are passed down to new cells, propagating the damage. The journey from a single mutated cell to a full-blown tumor is a complex and multifaceted one, involving a complex interplay of genetic and environmental factors. It's a journey that underscores the intricate workings of our cells and the delicate balance that governs our health. Imagine a cell preparing to divide, its DNA meticulously copying itself to ensure that each daughter cell receives a full set of genetic instructions. This process, while remarkably precise, is not immune to errors. Occasionally, a wrong letter is inserted, deleted, or duplicated in the DNA sequence, like a typo in a hastily copied text. These seemingly minor errors, known as mutations, can have profound consequences. Some mutations may be silent, having no noticeable impact on the cell's function. Others, however, can disrupt the delicate balance of cell growth and division. Certain genes, known as oncogenes and tumor suppressor genes, play critical roles in regulating cell growth. Oncogenes, when activated, act like accelerators, pushing the cell to divide uncontrollably. Tumor suppressor genes, on the other hand, act as brakes, keeping cell division in check. Mutations in these crucial genes can disrupt this delicate balance. An activated oncogene can lead to uncontrolled cell proliferation, like a car stuck with a jammed accelerator. In contrast, a deactivated tumor suppressor gene removes the brakes, allowing cells to divide unchecked. As these mutations accumulate over time, the cell's growth becomes increasingly dysregulated, paving the way for tumor development. While some genetic errors arise spontaneously during cell division, our environment also harbors a multitude of cancer-causing agents, known as carcinogens. These insidious actors can damage our DNA, increasing the likelihood of mutations that can lead to cancer. One of the most well-known carcinogens is tobacco smoke, a potent cocktail of chemicals that can damage DNA directly and increase inflammation, further contributing to genetic instability. Prolonged exposure to ultraviolet radiation from the sun or tanning beds is another significant risk factor. UV rays can penetrate the skin and damage DNA, leading to skin cancers like melanoma. But carcinogens aren't limited to smoking and sun exposure. They lurk in our environment in various forms, from industrial pollutants and air pollution to certain chemicals in processed foods and even some viruses. These agents, often acting silently and over long periods, can leave their mark on our DNA, increasing the risk of developing cancer. Protecting ourselves from these environmental hazards is crucial in reducing cancer risk. Simple measures like quitting smoking, using sunscreen, and making healthy lifestyle choices can make a significant difference in safeguarding our genetic integrity.